Good morning, ladies and gentlemen, colleagues. First, I would like to sincerely apologize for not being with you physically today in Seattle. An unexpected but important commitment has arisen that I just found out about literally a couple of days ago, which would necessitate my staying here in Washington and not being able to make the trip to Seattle. That's the bad news, that I can't be with you personally. The good news is that it involves people in the executive branch of the federal government at a much, much higher level than I am, and the discussions of a subject that's of common interest to all of us, namely global health. But having put in such a considerable amount of thought and effort into preparing this talk, I did not want to just not show up and not give the talk. So we took the opportunity to tape it and present it to you here today. So let's get on with the first slide and the talk itself. As you can see from this first slide, we've chosen the subject of the state of the science in malaria research. Now, I don't need to tell this audience, who is very familiar with the extraordinary disease burden of malaria, a disease that has been with us historically for such a long period of time, that still today, as you know the numbers as well as anyone, that every 40 seconds a child dies from malaria, a completely unacceptable situation. Having said that, we're very much heartened by the recent successes, and it's shown on this slide, the rollback malaria report from September of this year that heralded the successes that we've been experiencing already in the control of malaria in certain regions, and the anticipation and the hope aspirationally that we may have a near zero malaria death within a reasonable period of time, measured within hopefully this decade, if efforts are in fact sustained. So whenever I talk about research and research agenda, I always must appropriately tip my hat to the implementation of already existing tools which have had such a major beneficial effect. But as it said on the previous slide, we need to sustain this. And we all recall very clearly a few years ago where we had the call by Bill and Melinda Gates about the goal of eradicating malaria, because it in fact can be done. And in that discussion, there was clearly a place for the development of additional scientific breakthroughs that will be needed to truly end the scourge of this disease. In fact, we went right to it, and what was formed was the Malaria Eradication Research Agenda, discussions among people from many sectors that really identified actionable research that was based on global community input from a number of partners. What the bottom line is that although a number of research projects have led to advances that are already being implemented, in many respects, we really do need to bring malaria research squarely into the technical and other advances that we now have available to us in the 21st century. In other words, we really have to adapt to old and new malaria challenges, but we need to do it through a multifaceted research agenda, an agenda that makes use of technological advances that other fields of medicine and global health have actually utilized in many respects better than we have done in the field of malaria research. So I'm going to touch on each of these 6 areas of malaria research, and obviously I cannot give a comprehensive review of everything that's going on or planned, but I do want to point out a couple of salient features of each just to stimulate some thought in you. I spoke about bringing the malaria research agenda into the 21st century. Although genomics is not going to be the answer to all of the problems, the fact is that we have a solid genomic base for our malaria research and development, given the fact that all 3 players in the malaria issue have had their genomes sequenced and studied extensively. The human host, the mosquito vector, and the parasite itself. I'm going to return to certain aspects of this concept of genomic basis throughout this relatively brief talk. So let's start off with the issue of the host. it is very clear that the role of immune responses in malaria pathogenesis and protection, indeed to this day, after so many years of clinical experience, remains elusive. This slide, which shows the population indices of immunity in an endemic area related to the age of individuals, clearly tells us, bright and clear, that in fact, even though one is exposed to malaria, you do not develop lifelong protective immunity against reinfection. You certainly have an important impact on the severity of the disease, but particularly when one talks about the goal of eradication of malaria, the fact that even with the naturally acquired immune responses, you don't, in many cases, protect completely from infection, which really raises some fundamental questions in our understanding of malaria immunity. Some of these are shown in this slide. Why is immunological memory slow to develop? It's incomplete and short-lived. A very important question and an unsolved problem. What is the correlate of immunity at each of the complex phases of the parasite's life cycle? What are the mechanisms whereby the immune system controls severe versus mild disease? And which of the many antigens of the parasite induce responses that actually do confer protection? We're familiar with this various stages of potential immune mechanisms of the Plasmodium life cycle. I need not go over it with this audience, but merely to point out the differences, complexities, and in some cases overlap of the definable immune parameters that are operable at each stage of the life cycle. We're going to get back to, in a few minutes, the interesting and important immunological responses that occur actually at the liver stage, because this will have some relevance when we talk about the possibility of developing different types of vaccines related to protection against actual infection. Having said that, it's important to point out that there are indeed unique opportunities for human studies in malaria. In fact, opportunities that we may not have in other important diseases such as HIV, influenza, tuberculosis. For example, highly endemic areas can provide a setting for very efficient longitudinal studies, since most of the enrolled people in the studies will in fact develop malaria within a relatively short time frame. So the attack rate of the disease that we're measuring is extremely high, which really does provide a unique opportunity. Again, I know you know this, but it's important to point it out. Also important is the fact that challenge studies are not only possible, but they're ethically feasible in healthy volunteers. And this is predicated on the fact that pre-erythrocytic stages can be carefully examined with prevention and cure of disease in the subsequent blood stages merely by administering effective drugs. So we do have some interesting, unique opportunities for human studies related to the host itself. Also, getting back to the genomic basis of malaria research and development and the human host, the very fact that we have such a handle on the genomic makeup of the human host allows us to do studies that we had not been able to do just a decade or so ago. And that relates fundamentally to what we call the systems biology approach, particularly using the now available high-throughput technologies, such as the gene expression profiling, multiplex analysis of factors such as cytokines and chemokines that are triggered at various stages of parasitic disease, multi-parameter flow cytometry, and importantly, the availability of critical computational modeling. When one talks about the immune response to malaria in the host, one must address the innate immune response to malaria. Now, I know all of you know, and in fact, it's been brought to our attention most recently with the appropriate awarding of the Nobel Prize this year in physiology or medicine, to 3 scientists who were involved not only in the innate immune response, but also in the relationship between the innate and adaptive immune response, namely Bruce Beutler and Jules Hoffman for their work on toll-like receptors, and Ralph Steinman for his work on dendritic cells, which bridges the gap between innate and adaptive immunity. I bring this up because the understanding of the innate immune response and its relationship to adaptive immunity has become a very important issue with regard to malaria. And these 2 papers, which appeared a couple of years ago and most recently this year in PNAS, are good examples. And in these papers, it was shown that you could actually interfere with the activation of toll-like receptors. And this could be a strategy to prevent some of the deleterious inflammatory responses that mediate pathogenesis and disease severity. Again, a very important avenue of study in the host of this important disease. So let's quickly move on to the parasite itself. Again, an opportunity that we did not have years ago of being able to study very carefully the parasite at the genomic level due to the sequencing capabilities that are available to us. So with regard to the malaria parasite genome, it does indeed play an important role in understanding the spectrum of the disease. There are a number of very good papers. This one I just bring to your attention because it's something that I think people in other fields don't fully appreciate. We often talk about, and I did just a few moments ago, a systems biology approach to the human host. We now actually, because of the technological capabilities that we have, that we can actually understand the relationship between parasitic gene expression and clinical disease. In essence, a systems biology approach of the parasite itself. Moving on to the vector. Malaria vector control, there have been traditional approaches that we all are familiar with, many of which have been extraordinarily successful at times and then have failed at times. Insecticides, environmental modification, the very productive use of bed nets, most recently in the control and even elimination of malaria in certain regions. However, there are some novel approaches that are being implemented and will continue to be implemented. For example, biopesticides that target only the mosquitoes, various types of traps and feeding stations, novel chemicals or formulations that slow the development of resistance to the insecticides, and importantly, and really very exciting, is the use of natural parasitic fungi or bacterial symbionts to actually kill the mosquitoes or to affect their survival and transmission. Getting back to the genomic basis for malaria research, some novel approaches to malaria vector control are the utilization of transgenic mosquitoes, genetic modification that can actually impact survival or pathogen development and transition. Again, examples of our bringing the whole area of malaria research into the 21st century technology. This applies also to diagnostics. Talking about some of the current status of malaria diagnostics, clearly microscopy remains widely used. However, there has been recently the distribution and the use of rapid diagnostic tests. This has increased and they've become less expensive. They improve accurate diagnosis, they promote appropriate treatments, and they reduce expenses of malaria control. But we can actually do even better. We need better tools in the realm of diagnostics if we're really going to realistically look forward to the elimination and eradication of malaria. Because, for example, we need to improve and refine both the sensitivity and the specificity of these diagnostics, particularly and including in low incidence settings when you talk about ultimate eradication. They also need to be suitable to the field itself. It would be clearly advantageous if they were non-invasive. And you also have to consider other issues such as the detection of G6PD deficiency in patients, for example, with Vivax infection. A very interesting tool has been put forth and actually was published online just in the spring of this year, that students in a Microsoft-sponsored contest designed a smartphone application that can be used to actually remotely diagnose malaria. So what it really is, it's telemedicine, as it were, at its best in a field setting using technologies that are now becoming widely available in other arenas, now applied to malaria research. Therapies, again, clearly an important goal in the thrust of our modern research approach. Everyone is aware, and this is just one example of a publication that underscores this, of the extraordinary positive impact of the treatment with artemisinin-based combination therapy that has really transformed the therapeutic approach to malaria with an enormous impact on morbidity and mortality. However, as we all fear, and as happens almost invariably when you treat diseases over a period of time, it is becoming clear among us, and I don't think anyone doubts the data now, particularly the data coming from the Cambodian border, that artemisinin is beginning to lose its potency, and as it loses potency, it's the first sign of impending development of resistance. This has obviously not gone unnoticed by any of us in the field. The WHO has put out in January of this year a global plan for artemisinin resistance containment. This is a picture of their news release from January. They called for urgent action to protect the therapies of malaria. The NIAID, together with the Fogarty Center, had a meeting in November of 2010, which, as you can see from this flyer, was entitled Artemisinin-Resistant Malaria, Research Challenges, Opportunities, and Public Health Implications. From that was published a paper in the New England Journal of Medicine, in fact, that actually called for a comprehensive genome-wide search for the molecular basis of delayed parasite clearance that we were seeing, and the use of genome-wide association studies and assessing the signatures of recent strong selection is warranted, as was quoted in that paper. In fact, a very exciting paper was already in the process of being written, was published actually in August of that year from the NIH and other collaborators, which was really very elegant, because I'm sure most everyone in this audience is aware and saw this paper. It actually combines, and again, the 21st century approach of high-throughput screening with a genome-wide association, not of the host, but actually of the parasite itself. And in that study was identified a number of candidate antimalarial compounds, as well as the parasitic genes associated with response and resistance. A very good example of really bringing important areas of malaria research into the 21st century technology. Yet there are still some unmet needs for malaria therapeutics. We need drugs, improved drugs, to treat liver stage infection and prevent relapsing malaria. We need drugs to kill the gametocytes. And finally, on my list of 6 is the issue of vaccines. Again, you're all familiar is with the complexity of the life cycle of malaria comes the complexity of immunological response and opportunities and challenges for the development of vaccines, as shown on this slide. Depending upon the slide, the major thrust of a vaccine objective could be either to prevent infection, to prevent clinical disease, or to prevent transmission. There have been a number of important, some quite successful thus far, approaches in the development of malaria vaccine candidates. For example, the use of subunit vaccines. On this list, you see the RTSS. I'll get back to that in a moment. A variety of vectored vaccines using the vector technology and combined modalities, for example, heterologous prime boost. Another exciting area is the use of whole parasite vaccines, such as irradiated sporozoites, which I'll again get to in just a moment, genetically attenuated parasites, infection-treated vaccination regimens. Again, I'll show you an example of that, in which you vaccinate with a live attenuated and then actually treat the individual. Let's just mention RTSS vaccine, which is the furthest advanced in development. You're going to be hearing or have already heard more about this. The phase 2 studies were really quite encouraging, with about a 50 to 60% protection against disease. The ongoing phase 3 clinical trial, the results of which are really imminent, a large trial involving 16,000 children. Every one of us in the field look with great anticipation to the results of this important trial. Now, the idea of sporozoite inoculation is really quite interesting. There was an important study a couple of years ago using this whole parasite-based approach, where one interrupted the parasite life cycle after the liver stage with chloroquine treatment, allowing the person to be infected with the sporozoite but not get disease, and showed that actually there was 100% protection that lasted for approximately 2 years, which is suggesting something very important of protective immunity that targets the liver stage. In this regard, there was a very interesting paper just last month from Steve Hoffman and his colleagues, which appeared in Science. There was some sobering news in there and some rather encouraging news that's triggering a considerable amount of thought about different types of approaches. The first part of it showed that the phase 1 trial of the purified attenuated sporozoites, if delivered intradermally or subcutaneously, really gave very low levels of immunogenicity and protection. However, the use of an alternative delivery, namely intravenously, in non-human primates elicited very potent liver CD8 positive T cell responses. Now, with all the caveats associated with the technicalities involved with IV delivery, the idea that you can actually induce potent CD8 responses, and again, with all of the weaknesses in some respects of a mouse efficacy model, the IV delivery showed dramatically more protection than intradermally. The proof of concept study in humans to actually just ask the question, would IV delivery really induce in human the potent types of CD8 responses that we have seen in non-human primates, and will that be associated with protection against challenges? I'm happy to say that these studies have begun in Bethesda just this past month, and the third patient was actually injected just a couple of days ago. So this is just a heads up to keep our eye on something that might be really a very important proof of concept. I do want to mention the NIAID-funded International Centers of Excellence for Malaria Research, the ICEMERs. These are 10 multidisciplinary centers with studies in 20 countries. And the reason I mention it, because it really bridges the laboratory to the field and vice versa. What it does is something very important when you're dealing with a disease like malaria. It addresses malaria in different transmission settings, namely looking at it within the context of where it occurs and bring the fundamental basic and clinical sciences into the field. I just wanted to mention that because we really have a good deal of hope that this is going to bring some very important advances, or at least some important ideas on the table to be further pursued. So I'd like to close by just showing you the title of a paper that Lee Hall and I wrote a couple of years ago regarding malaria control, elimination, and eradication, where we discussed the role of the evolving biomedical research agenda. And I want to quote one of the points that we made in that paper, and that is that the final challenge is to ensure the vigor and sustainability of the substantial biomedical research effort that will be required to eliminate and ultimately eradicate malaria. So I want to underscore the importance of malaria research being one of the many components that will have to be put into the effort of ultimately eliminating and eradicating malaria. It is one component, but a very important component, and I promise you, this audience, that we will continue to pursue this with great vigor and enthusiasm. And so again, I want to once again apologize sincerely for not being here with you in person and having to use this kind of technical approach. But I really felt strongly that I wanted to address you and to impart upon you my ideas, the putting together of what we have been discussing with so many of you in the audience, and to reinforce my enthusiasm for what I consider one of the most important problems in global health, and something that we will continue to address very robustly and with considerable enthusiasm. Thank you all very much.